Hello everyone. Today I will discuss DNA, chromosomes and genomes. Information about DNA is shown in the video about Chapter 1 of Albert's The Cell. Links below. Genomes are all the genes of the chromosomes of an individual. The genetic information is stored in DNA and contains the information for proteins. In eukaryotes, the DNA is divided in chromosomes. A chromosome contains a very long linear DNA molecule. Every cell contains 2 copies of each chromosome, one from the mother and one from the father. These are called homologous chromosomes. Non-homologous chromosomes pairs are the sex chromosomes. So each cell contains 46 chromosomes, 2 copies of 22 homologous chromosomes and 2 sex chromosomes. In a gene you have introns and exons. Exons contain coding sequence and introns contain non-coding sequence. Although a lot of people doubt about the non-coding sequence in introns. DNA is too long to be unwrapped in a nucleus, therefore it is wrapped around histones. When DNA is around a histone, it is called a nucleosome. Nucleosomes are zigzagged or solenoid and form chromatin. Many repetitions are called chromatin fibers. Both models are supported with experiments. At the moment it is not yet known if they zigzag or solenoid, or do both. One thing that is important for a nucleosome is the linker histone H1. It is between the 2 DNA strands. The linker is important because it prevents unwrapping of the DNA strands around the histones. Nucleosomes under a microscope can be seen as beads on a string. Every nucleosome is separated with a linker DNA. That is a piece of DNA that sits between the 2 nucleosomes. A histone consists of 4 core domains, H2A, H2B, H3 and H4. The assembly of an histone is as follows. First, 2 separate H2A and H2B dimers are formed, and secondly, 2 separate H3 and H4 dimers are formed. Now, H3 and H4 dimer combine and form a tetramer. 2 H2A, H2B dimers bind to the tetramer and an octamer is formed. The DNA wrapped around the histone is 147 nucleotides long and is wrapped in 1.7 turns around the histone. Between the DNA and the histone, many hydrogen bonds are formed, to be precise 142. This is why the DNA is so tightly wrapped around the histone. Half of the hydrogen bonds is connected to the DNA backbone. DNA has a preference to have adenine and thymine bases on the histone. Therefore, cytosine and guanine are on the other side. Every core histone has an N-terminal amino acid tail. These tails are subject to different types of modification to control the chromatin structure and function. I will discuss this later. ATP-dependent chromatin remodeling complex can bind the core of the histone and opens the DNA. It remodels the histone by removing only H2A, H2B dimers or by removing the whole histone. It is replaced with new parts. This is more complex than it sounds because it all has to do with epigenetic memory. There are 2 forms of chromatin, heterochromatin and euchromatin. Heterochromatin has a dense structure and thereby mostly inactive in transcription. It can open the chromatin its formation, whereby it becomes euchromatin. Euchromatin is transcriptionally active because it has an open structure. Transcription factors are now able to bind to the DNA. Heterochromatin and euchromatin are formed by histone modifications on the tails of the nucleosome. These modifications cause inactivation or activation of the chromatin. There are 4 forms of modification: acetylation, methylation, phosphorylation, and ubiquitination. Some examples are methylation of lysine 9 causes gene silencing, so heterochromatin. Acetylation of lysine 9 and methylation of lysine 4 causes gene expression, so euchromatin. Due to phosphorylation of serine, lysine 9 cannot be methylated because the size of the phosphor is too large, whereby methyl cannot fit next to it. Later I will discuss more modifications and their purpose. The tails of the histones are named after the core, so there's H2A, H2B, H3 and H4 tails. There are always 2 tails. The example I just described are modifications to H3. Acetylation is done by histone acetyltransferase, in short, HOT. It binds to acetyl group to the positive NH3 group. It becomes an NH group and it is not charged anymore. To remove the acetyl group, you need an histone deacetylase, HDAC. Methylation is done by histone methyltransferase, in short, HMTase. SAM is involved in this reaction and CH3 is removed. It is put on the molecule that needs to be methylated. There are 3 different stadia of methylation. You can have 1, 2, or 3 CH3 groups attached to the molecule. The function is depending on the amount of CH3 groups attached. Histone demethylase, in short HDMase, remove CH3 groups. The core histones have some variants. The most important variants are H3 variant, CENP-A and H3.3, H2A variant, H2AZ, macro H2A and H2A-BBD, H2B variant, H2B-FWT. I will discuss CENP-A. Chromosomes are attached to each other via cohesin. In order to divide the cell, the chromosomes need to be divided. To pull the chromosomes, you need a kinetochore. Centrosomes form microtubules and they will attach to the kinetochore and pull. The kinetochore is attached to the chromatin. On the side of the kinetochore, the chromatins contain CENP-A histones. Below are normal H3 histones. CENP-A is a nucleosome that is specific for centromeres. It contains a specific sequence, the alpha satellite. An alpha satellite is a short sequence that repeats. Due to this specific sequence, CENP-C is bound. The function is to make the connection between kinetochore and centrosome. When the chromosome is pulled apart, the chromatins will stay on their place. CENP-A contains less DNA than a normal H3 histone. There is less DNA coiled around the histone because it has a slightly different structure compared to H3 histones. It misses a small part of the alpha helix, whereby it cannot bind to the entire DNA. As last, I will further discuss the histone modification and their purpose. Histone modifications will be determined if a gene is active or inactive. We have transcriptionally active marks and transcriptionally inactive marks. This can be acetylated, methylated, phosphorylated, or ubiquitinated. As you can see, there are many forms. All these modifications are done via specific modificators. For example, H3K27 is methylated by EZH2. So in addition to the information of the DNA, the histone modification adds a second layer of information. This is called the histone code. Genetic and histone code together provide the information if and how genes should express. There are differences between genetic and histone code. Histone codes are reversible, genetic codes are not. Specific partners of modifications influence binding affinity of other regulated protein to the histone tails. Proteins that recognize specific modifications are the bromodomains. They recognize acetylated lysine. Chromosomes recognize methylated lysine, and pseudodomains recognize methylated lysine. There is a crosstalk between modifications. Histone modifications influence each other. For example, acetylation of H3S10 will disable the ability to methylate H3K9. The consequences of histone modifications are change in the chromatin structure, recruitment of specific proteins, and repulsion of specific proteins. The type of modifications: acetylation, methylation, mono, D or 3, phosphorylation and ubiquitination, and the position of the modified amino acids in the histone tails determine which proteins will bind. As you can see in this picture, I made a map of methylated H3. You can see there's already a lot of things in it, and it's just a small part. So you can see that the epigenetic information is very important. It is just not a little thing, it's immense, it's huge, it's very important. As you can see here, there are loops back. H3K9 activates HP1, and then SUVAR 3 to 9 is activated, and it methylates H3K9 again. So you can see that it's a circle. For example, H3K79, demethylation is not possible. Dot 1 methylates H3K79, and if it's methylated and it cannot demethylate, it can cause leukemia. Of course, further research is necessary, but that's what they suggest at the moment. Take H3K4. There are 4 things that can methylate H3K4. But this is just methylation. H3K4 can also be acetylated. The same is for H3K9. There are so many forms, and everything interacts with each other. So don't forget about the epigenetics, because it's more important than you think. So this was it. Thanks for watching, and I hope to see you in the next video. Bye!